Hi everyone, my name is Mandolin McGaw, and today I will be presenting on the impact of systemic antibiotics on the human skin microbiome and antimicrobial resistance. This presentation is based on a study by Jae-Hyun Jo et al., which explores how systemic antibiotics alter the skin microbiome and contribute to antimicrobial resistance. Let's dive in. The key takeaway from this study is that systemic antibiotics cause long-term shifts in skin microbiome composition, often leading to an increase in antibiotic resistance. Unlike the gut microbiome, which can recover relatively quickly, changes in the skin microbiome can persist for over 200 days after antibiotic use. All right, now let's talk about what makes this relevant. Over 200 million outpatient antibiotic prescriptions are written annually in the United States alone. The majority of patients who are seen at outpatient facilities leave with antibiotic prescriptions, a third of which are entirely unnecessary. Misuse and overuse of antibiotics fuel the development of antibiotic-resistant bacteria, a major public health concern. This is because antibiotics kill off susceptible bacteria, leaving just the resistant bacteria with plenty of resources to grow and multiply. Natural selection is what guides the emergence and spread of resistance genes. Spontaneous mutations lead to the appearance of resistance genes, and then these are transferred between bacteria. Vertical gene transfer results in spread from parent cell to offspring, while horizontal gene transfer allows for genes to be transferred to different species. Commensal bacteria can acquire resistance genes and transfer them to harmful pathogens, a well-documented phenomenon in the gut. For example, extended spectrum beta-lactamase producing bacteria were found in the digestive tracts of 12 out of 18 students after returning home from India. All of these students had baseline testing, and no ESBL bacteria was found prior to their travel. This has also been observed with quinolone resistance in travelers from Southeast Asia. So we're going to take everything that we just talked about and we're going to apply it to the skin microbiome now. The skin microbiome impacts health in a multitude of ways. Microorganisms produce antimicrobial compounds and maintain the acidity of the skin, which prevents pathogen invasion. The skin microbiome also stimulates the immune system by priming T cells, interacting with pattern recognition receptors to maintain defense mechanisms, and reducing inflammation. All of this is an essential part of wound healing and infection prevention. Byproducts of microbial metabolism also contribute to the function of the skin barrier. Lastly, the needs of different niche environments are promoted by their unique microbiomes and their specific metabolic byproducts. Resistance in the skin microbiome is not as well studied as the gut microbiome, and until recently, there was almost no available data on the topic. A recent study did identify Staphylococcus species as a resistance reservoir, which is concerning given that it is a common skin commensal. This same study also identified fluoroquinolone, tetracycline, and cephalosporin resistance genes as the most common. Now that we have that background information, we're going to dive right into this study. It is called Alterations of Human Skin Microbiome and Expansion of Antimicrobial Resistance After Systemic Antibiotics, and it is by Jae-Hyun Jo et al. In 2021. Before we start talking about the details, let's first establish the objective of this study. Systemic antibiotics play a crucial role in treating infections, but their widespread use has raised concerns about their impact on the human microbiome. While much research has been dedicated to studying antibiotic effects on the gut microbiome, there is limited knowledge about how systemic antibiotics alter the skin microbiome, which serves as a critical barrier and defense system for human health. The primary objective of this study was to investigate the impact of different systemic antibiotic regimens on the human skin microbiome. Specifically, researchers aimed to determine how antibiotics influence the composition and diversity of the skin microbiome over time, whether these changes lead to an increase in antibiotic-resistant bacteria, potentially contributing to antimicrobial resistance, how long-lasting these effects are, particularly in comparison to known disruptions in the gut microbiome, and what clinical implications this data presents. Now that we understand the objective of the study, let's explore the researchers' hypotheses. Given the well-documented effects of systemic antibiotics on the gut microbiome, the researchers hypothesized that systemic antibiotic use would also disrupt the skin microbiome, leading to significant changes in microbial composition and an increase in antimicrobial resistance. Specifically, they predicted 3 things, the first of which revolves around microbial disruption. They predicted that individuals receiving 100 milligrams of doxycycline and TMP-SMX would experience the greatest decrease in microbial diversity, with certain bacterial species becoming dominant while others diminish. They also had a prediction revolving around emergence of antibiotic-resistant bacteria. They predicted that the stress imposed by antibiotic exposure would select for resistant strains of bacteria, which would allow them to proliferate within the skin microbiome. Finally, they predicted that unlike the gut microbiome, which is relatively dynamic, the skin microbiome would take longer to recover from antibiotic exposure, possibly leading to extended shifts in microbial communities. By testing this hypothesis, the researchers aimed to shed light on the extent to which systemic antibiotics impact not just the bacteria that they target, but also the broader microbial ecosystem of the skin. To test these hypotheses, researchers designed a structured study to examine the effects of systemic antibiotics on the human skin microbiome. This study was a prospective, randomized trial that recruited healthy volunteers. Participants were divided into groups based on the regimen they received. The 4 regimens tested were doxycycline, 200 milligrams or 100 milligrams. This is a commonly prescribed tetracycline antibiotic used for infections and inflammatory skin conditions. They also used cephalexin, which is a first-generation cephalosporin frequently used for skin and soft tissue infections. They used trimethoprim and sulfamethoxazole, also known as TMP-SMX, and this is a combination antibiotic that targets a broad range of bacteria. There was also a control group that received no antibiotics that was used as a comparison group to observe natural variations in the skin microbiome over time. Each participant underwent multiple microbiome sample collections before, during, and after antibiotic treatment, with follow-up extending up to 1 year to assess long-term effects. To analyze the changes in microbial communities and antibiotic resistance, the study used advanced genomic sequencing methods, including shotgun metagenomic sequencing to identify all microorganisms present and assess microbial diversity, and whole genome sequencing to focus on specific bacterial strains and detect antibiotic resistance genes. By designing the study this way, researchers ensured a comprehensive approach to understanding how systemic antibiotics impact the skin microbiome. Now we're going to take a closer look at the specific methods used in this study. We're going to start out by talking about whole genome sequencing. This technique focused on specific bacterial strains, allowing researchers to take a deeper look at individual microbes. It was particularly useful for antibiotic resistance analysis as it identified genetic mutations and resistant genes that emerged after antibiotic exposure. This slide provides a general overview of how WGS works. First, DNA is extracted from the source, which would have been the bacteria cultures in this study. The DNA is then fragmented, barcoded, and amplified to prepare it for sequencing. While on the sequencing instrument, large numbers of sequencing reactions occur in parallel, which generates DNA that is aligned to a reference genome. Shotgun metagenomics actually works very similarly to whole genome sequencing, except that DNA is extracted from all the different organisms in the community in one sample. Individual organisms are then separated during sequencing preparation and individually sequenced. This method allowed researchers to identify all microorganisms present in the sample, including bacteria, fungi, and viruses. It provided a broad overview of microbial diversity and how different species changed over time. Additionally, the data from this technique was used to calculate diversity indexes, which help measure how much the microbiome shifted due to antibiotic exposure. So you might be wondering why we would need two different sequencing methods here. We're going to compare the methods and discuss why we actually needed both. So shotgun metagenomics is useful for identifying all organisms present in a sample. This data can be used to perform diversity indexes, and it can also provide some information about genetic makeup of individual organisms. So shotgun metagenomics was used to identify certain bacteria that were worth looking further into with whole genome sequencing, which is useful for taking a deeper look at a specific organism, and it was used for the antibiotic resistance analysis. Combining these approaches ensured that the study could capture both broad trends and precise genetic changes occurring within the microbiome. With these advanced sequencing tools, researchers were able to uncover significant findings about how antibiotics disrupt the skin microbiome. Let's move on to see what they discovered. So what exactly did researchers discover? We're going to move on now to talk about the results of the study. What did researchers actually discover about the impact of systemic antibiotics on the skin microbiome? Figure 1b shows the Bray-Curtis dissimilarity of the microbiome over time, comparing individuals on antibiotics, which are the colored lines, with untreated individuals, which are the gray lines. Bray-Curtis dissimilarity measures how much a microbial community has changed over time. Higher values indicate greater microbial shifts, while lower values show a more stable microbiome. So if you look at these graphs on the right, the top one is doxy 20, and then under that is doxy 100. These are milligrams, and then cephalosporin and TMP SMX. The x-axis is number of days, and the y-axis is the dissimilarity to day 0, which is the Bray-Curtis distance. So the higher the number on the y-axis, the greater the Bray-Curtis value is. And as you'll see for each graph, the colored lines have significantly higher Bray-Curtis distance values than the gray graphs. The antibiotics doxycycline 100 milligrams and TMP SMX cause the most severe and prolonged disruptions to the skin microbiome, and you can see that here too. Doxy 100 milligrams again is the red lines, and TMP SMX are the blue lines. These changes persisted for over 200 days, much longer than the disruptions typically seen in the gut microbiome. And the skin's microbiome, which is normally stable, took a long time to recover after antibiotic exposure. Even for the other antibiotics, doxy 20 milligrams and cephalosporin, their Bray-Curtis distance was still high even after about 50 to 100 days, which is what is normally seen with a gut microbiome. An increase in antibiotic resistance was observed in Staphylococcus epidermis. This tracks with what we talked about in the background information, where resistance has been seen previously in Staphylococcus species. specifically 2 doxycycline resistance genes, TET-K and TET-L, and 2 TMP SMX resistance genes, DFRC and DFRG, were seen. Even after treatment finished, this resistance continued to be seen. Doxycycline resistance was seen up to 336 days, which is when the last samples of the study were taken. TMP SMX resistance followed a similar pattern. The graphs on the next slide will show this in more detail. These graphs show relative abundance by number of days. Relative abundance is on the y-axis and number of days is on the x. The different colored lines are the different serotypes of Staphylococcus epidermis, and the type of line shows the level of resistance. Dotted lines show that they were susceptible, dashed are intermediate, and solid are resistant. Across the board, meaning with each patient 5 through 8, you see a rapid increase in intermediate and resistant S. epidermis samples. Many of these were still abundant at the end of the study despite decreasing. For example, with subject 7, even though resistant bacteria started to decline about halfway through, they still had an abundance higher than some of the susceptible bacteria, especially for AC. Some even started to increase again towards the end, like with subject 8, and also subject 6 if we're talking about intermediate bacteria. Another major finding of this study was that bacterial stress increases horizontal gene transfer. Researchers found an increase in transposable elements, which are genetic sequences that help bacteria transfer resistant genes to one another. This suggests that antibiotic exposure triggers stress responses in bacteria, leading to greater gene exchange and the potential spread of resistance. This could lead to an increased spread of antibiotic resistance genes, which is clinically very important. You can see this represented on these graphs to the left. Panel A is the doxycycline 100 milligram group. This graph compares the relative abundance of metagenomic gene clusters at day 0, which is pre-antibiotic, and day 56, which is post-antibiotic. The x-axis represents the log transform mean abundance of MGCs at day 0 and day 14, while the y-axis shows their abundance at day 56. Hexbin density plots, which are blue to dark blue in color, indicate the concentration of MGCs. The lighter the blue, the more abundant. The red points highlight MGCs that significantly increased or decreased. Panel C is the TMP SMX group. Similar to Panel A, this figure shows MGC changes between day 0 and day 42. Over 50% of the increased MGCs contained GO terms for DNA integration and mobilization, confirming that TMP SMX treatment also enhanced genetic exchange among skin microbes. Throughout this presentation, we have talked about how the skin microbiome recovered slowly, but it's very helpful to have something to compare this to. That's why it's so wonderful that this study also looked at the gut microbiome. This slide goes and compares the recovery of both. It was seen that the changes in the skin microbiome did last longer than what's seen in the gut microbiome. And this is likely because the skin has stable niches while the gut tends to be more dynamic. This is demonstrated by panels D and E, which show the Bray-Curtis dissimilarity of the skin and gut microbiomes. Panel D shows the skin microbiome stability, and even after 200 days, you can see that the skin microbiome remains significantly altered. Most of the samples are about 0.5 on the Bray-Curtis scale at this point, maybe a little above or below. Panel E shows the gut microbiome stability, and here you can see a steep decline in disruption around day 50, indicating that the gut microbes bounce back more quickly than the skin microbes. and around day 50, the majority of the values fall around 0.25, a little above or below. Now that we understand the key findings, let's discuss what they mean in a clinical setting. How can we apply this knowledge to patient care? This study reinforces the need for careful and responsible antibiotic prescribing, both in treating infections and chronic diseases and conditions. Overuse of antibiotics not only disrupts the microbiome, but also promotes antimicrobial resistance, making future infections harder to treat. Certain conditions, like acne and rosacea, are commonly treated with long-term antibiotics, sometimes for months or even years. Given the prolonged impact on the skin microbiome, alternative treatments such as topical antibiotics, probiotics, or non-antibiotic anti-inflammatory therapies should be considered when appropriate. Even the low-dose doxycycline, which is generally being considered safe to use as an acne treatment, disrupted the microbiome. All of that being said, physicians should be aware of the potential for resistant skin infections following systemic antibiotic use. Future strategies could include screening for resistant bacteria in high-risk patients and developing targeted treatments that minimize microbiome disruption. What future improvements could be made on further research as well as this field in general? These results highlight the importance of replacing antibiotics with alternative treatments, especially in the case of chronic skin conditions where there is less risk associated with trialing a new method. If you were to trial a new treatment method on somebody that needed antibiotics because they had an active bacterial infection, you face the very real risk that they could get worse, have lifelong consequences, and even pass away. However, with conditions that are generally nuisance conditions, like acne or eczema, you don't face such catastrophic risks. There is still a lot of mystery surrounding antibiotic resistance in the skin microbiome, and the results from this study show that there is an urgent need to investigate it further, as well as the effect of antibiotics in the long run. To wrap up, let's highlight the key takeaways from this study. First, systemic antibiotics significantly disrupt the skin microbiome with effects lasting over 200 days, which is much longer than disruptions seen in the gut, over twice as long. Antibiotic resistance increases after treatment with commensal skin bacteria, particularly Staphylococcus epidermis, acting as reservoirs for resistance genes. Bacterial stress also promotes horizontal gene transfer, potentially accelerating the spread of antibiotic resistance. Future research is needed to explore alternative treatments, microbiome recovery strategies, and the long-term effect of antibiotic exposure. This is essential for improving antibiotic stewardship, which is necessary to prevent a shift from a healthy microbiome to a disrupted one that could put the patient at risk for future resistant infections. Lastly, no study is without fault. While this study provides valuable insights, it's important to acknowledge its limitations. The first thing that really stood out was the small sample size. With only 14 participants, the findings may not be fully generalizable to a larger population. And this is something that the authors actually expected. At the beginning of the study, they said they expected about a 50% dropout rate, so they had 20-something people originally. My suggestion for a future study would be to enroll 50 to 100 people, maybe even more. There is also a limited follow-up duration. The study tracked changes for 336 days, but we still don't know when or if the microbiome fully recovers. During the last sampling, many patients still had microbiome disruption. The antibiotic selection here was pretty narrow. Only 3 antibiotics were studied, meaning the effects of other commonly used antibiotics remain unknown. For example, quinolone antibiotics resistance was seen very commonly in skin bacteria, and they were not used in this study. Another one is genetic presence versus expression. Just because the resistance genes were identified doesn't necessarily mean that they were active. Future studies, including metatranscriptomic analysis, might actually help with this one. Recognizing these limitations helps guide future research and improves our understanding of how systemic antibiotics impact the skin microbiome. Thank you so much, everybody, for your time. I appreciate you listening. If you also find this topic super interesting or you have any questions, you can always reach out to me on Canvas or send me an email at my UFL email. This topic is particularly interesting to me because I was on doxycycline for acne growing up. So I'm always eager to hear more about it, to talk about it, to ask questions, to be asked questions. So please reach out if you have anything you want to say. Here is the first page of my sources cited. And finally, the last page. Again, thank you so much, and if you're still listening at this point, what are you doing? All right, bye everyone, thank you.